Section 2 of The Psychic Life of Micro-Organisms by Alfred Binet. This is a LibriVox recording. All LibriVox recordings in the public domain. For more information or volunteer, please visit LibriVox.org. Recorded by Leon Harvey. Chapter 2. Nutrition. After studying the organs, let us pass to a study of their functions. It is not our intention to devote special chapters to irritability, instinct, memory, reasoning, and the powers of evolution in microorganisms. This would lead to diffuseness of treatment. Our method will be quite different. We shall describe as a whole all the different manifestations of psychical activity attendant upon the actions of microorganisms in the exercise of the important functions of their existence. The present chapter will be devoted to psychical phenomena connected with the act of nutrition. All living matter possesses the power of continually increasing its mass by the inward reception of materials, and of simultaneously decreasing the same through the combustion of its substance with the oxygen of the atmosphere. The first of these processes is called nutrition, and the second respiration. We shall first examine the psychical phenomena which precede and determine the act of respiration. These phenomena are often very simple and of little significance. If the microorganism lives in the water, which is most frequently the case, the oxygen contained in solution therein passes directly through the cellular cuticle by dialysis and comes in contact with the body of the protoplasm, in which case the process of respiration is solely a chemical phenomenon. But it may happen that a minute organism chances into a medium containing little or no oxygen gas. Amid these new conditions, where it becomes necessary to move towards sources emitting oxygen by voluntary effort and directed motion, it has been discovered that a great number of microorganisms, and particularly bacteria, are capable of detecting the expansive power exerted by oxygen in the liquids in which they are found. When bacteria of putrefied matter are put in a drop of water containing no oxygen, but in which have been placed chlorophyll algae, or green euglena, or grains of chlorophyll obtained by crushing green cellules, nothing happens in the first instant. But if the preparation be illuminated so as to allow the chlorophyll to act, the bacteria are seen to exhibit very rapid movements and to proceed, altogether, towards the points of the preparation where the generation of oxygen is taking place, that is to say, about the grains of chlorophyll. Under these conditions, a chemical exchange is instituted between the chlorophyll and the herbaceous bacteria. The bacteria disengage carbonic acid gas and absorb oxygen. The chlorophyll fastens upon the carbon of the acid and sets the oxygen at liberty. If the preparation be darkened, the bacteria cease assembling about the chlorophyll grains, which, hid from the light, cease to disengage oxygen. The clustering begins anew if a ray of sunlight is again let touch the chlorophyll. Analogous facts have been observed under circumstances somewhat different. In the preparation from the intestines of the silkworm, M. Balbiani has seen bacteria which were uniformly distributed throughout all points of the preparation, gather about the green and undigested cellules of the leaves contained in the intestines, and bury themselves in them as if to partake of them. In other instances, the same naturalist has observed that bacteria developed in a drop of silkworm's blood would gather after a while about the globules of the blood, undoubtedly for the purpose of seizing the oxygen being absorbed by them. Upon the basis of these facts, M. Engelmann has established the method called the bacteria method. He regards bacteria as a living reagent which enable us to reveal the trillionth part of a milligram of oxygen, that is to say, a quantity scarcely greater according to the calculations of physicists than a molecule. This curious method enables us to explain biological problems, and which had hitherto remained unsolved. Before this, it was not known whether the colourless protoplasm of green plants could or could not disengage oxygen. It is now known, thanks to the bacteria, that grains of chlorophyll are the only points about which the liberation of oxygen takes place. The same method has enabled us to prove in the variegated plants that the maximum liberation of oxygen coincides with the maximum absorption of light. Thus, in the case of green algae, the red and the violet colours of the spectrum are the spots where the bacteria accumulate the thickest. Consequently, there is where the liberation of oxygen is greatest. Now these colours correspond to the lines of greatest absorption in the spectrum of chlorophyll. In the case of brownish yellow cellules, the maximum action is in the green. In the case of bluish green cellules, in the yellow. In the case of red cellules, in the green. The author has concluded from this that there exists a series of correlating substances which, like chlorophyll, have the power of dissolving carbonic acid gas. He calls them chromophylls. In the same way, moreover, this method enables us to solve the question of the distribution of energy in the solar spectrum. As M. Engelmann has remarked, it is interesting to see the bacteria come to conform our theories as to the composition of solar light. Bacteria are not the only organisms that eagerly make towards points where oxygen is to be found. A large number of other microorganisms act in the same way when they happen into a medium lacking oxygen. M. Ranvier has noticed that if a preparation containing leucocytes, screened from air, be examined for a certain length of time, the cellules will be seen to throw out long filaments towards the parts that faces the air side of the preparation. It appears then that a rudimentary oxygen sense exists in the protoplasm of proto-organisms. This sense does not merely appraise the organism of the presence of oxygen, it enables it, further, to gauge the tension, expansive power of the gas, so that when the tension becomes too powerful, the organisms are seen to flee before it. End of section 2.